And now we're going to transition to Dr. Olga Zhaxybayeva, who is an associate professor of biological sciences and an adjunct associate professor of computer science at Dartmouth University. Dr. Zhaxybayeva's research aims to understand how microbes change over time, and she'll be discussing the genomic clues to the origin, evolution, and diversity of life. So without further ado, everyone, please welcome Dr. Zhaxybayeva.
Hi. Hi, everyone. First of all, thank you for inviting me. Okay, now I can share the screen. I was not allowed to share screen before. I'm really impressed with Diana's efforts to make this event happen. Do you guys hear me okay? Diana, can you?
Yeah,
okay.
Sounds good.
All right, sounds good. And then you guys see my slides, is that correct?
Yeah, we can see that.
Okay, great. Just, making sure that we are good on technical side. All right. And the other thing that I've done, depending on how your stuff is set up, you can, whoops, I don't know what happened there. You can put my video into the upper right corner here. I tried on my slides to leave that space completely open. So what I decided I'm going to talk to you today, I know my title is a little generic, is about, in general, how I got to do what I'm doing now, and highlight just one project that is going on that we're really excited about and been producing some interesting results. So just to go, let me also change, sorry, there's a bit of a setup here. Just to go back to my high school time, I have very fond memories of my high school, which was a special school. Was kind of geared towards physics and math education, but of course, we had all other subjects, and I was really undecided what I wanted to do in the future. Had no idea. I actually didn't like biology that much. The way it was taught to us, it just didn't. It didn't click to me. I really like nature, but I didn't like it as what I wanted to study. But it was interesting. I really like coding, so computer science at that time wasn't called that way. Was really interesting to me. I like chemistry. I like physics, math. I like geography a lot. I like geology or sciences, but I really wasn't undecided. And so I was good at math at the time. So I just said, all right, well, I'll go to college to study applied mathematics this way. I can also do some coding, and we'll see how where I get there. So how did I end up being a professor in biological sciences? Well, turns out when I was in college, we had an optional seminar where a professor, he was in the physics department, came to talk to us about life in the universe. And he was talking to us about how vast the universe is. And, just to illustrate, I'm going to show you a picture that was taken by Voyager 1, which is the probe that was launched back in 1977. And when it was about 4 billion miles away from Earth, it sent back this picture. And most of the picture that you see here, this color pixels, are just noise from the camera. The only signal here is this little dot. And this pale blue dot is Earth. Everything else is pretty much empty space. And so in that lecture series, we spent a lot of times talking about how did life originate on Earth and how widespread is it in the universe? Kind of unknown questions, still unknown questions. And this is what was one of my turning points where I said, oh, this is so exciting. I should really do this. And moreover, the professor told us, well, you guys are math majors. You can work in this from mathematical computational perspective, because to really understand life, we need to understand information that creates life. And every living cell has information. Whoops. And this information is encoded in our genome. which we can define as a complete set of genetic material that is present in every cell, including every cell in your body. And so here it is. This is just a small portion of human genome sequence. Lots of this DNA bases, T, C, A, and G, that we can computationally just encode as letters, 4 letters, and somehow this stretch of information has instructions to create a human or any other living organism. So we're still trying to understand how we go from this code to a living organism, and understanding this will allow us to understand how life originated on Earth and elsewhere. And so this information is inherited. It's passed from one generation to another. You got your DNA from mother and father during this fusion of 2 gametes. Now bacteria pass information by duplicating the genome and then passing it to daughter cells during the cell division. And during this information transfer, there are DNA copying errors. And so it's actually not that frequent that they occur, but when they occur, they changes create these modifications that are descendant to the next generation, and that's what drives evolution, and that's what drives diversification of life as we know it. Now, what then we can do over time, we can use this accumulated errors or accumulated changes, now they're no longer errors, and we can go back in time. In a way, we can do archaeology of genomes. And I'm going to read this quote because I really like it, by Eric Lander. Evolution is, so to speak, an experimentalist that has been running experiments for 3.5 billion years since the origin of life on Earth. And wonderfully, the genomes of today's organisms retain the lab notes of these experiments so that we can go back and reconstruct the events that took place. And so just to illustrate it graphically, we have a present-day organisms. They have some genomes. They have this information. We can compare their genomes and we can track back the events that led to their diversification. I'm going to talk a little later about just kind of an overview of how we can do that. And hopefully, you know, we can go deep enough in time that we can, you know, peek at the early evolution of life, maybe to the origin of life, but at least to some early evolutionary events that took place. Now that's all really cool and exciting, and I was really, as an undergrad, was really excited about this. And then I had a chance when I was 18 to meet a Nobel Prize winner, Professor Manfred Eigen. who he got a Nobel Prize in chemistry, but he spent a pretty big chunk of his career studying the origin of life question. He said, well, Olga, yeah, it's really exciting. These are the important questions. But he said, you know, these are tough questions. And as a young scientist, you probably want to start small. And maybe you want to start, you know, identifying some smaller problems that kind of fit within the framework of understanding evolution, but maybe not directly address the origin of life question. And so the rest, as I say, is history. So I spent my graduate school years working in the molecular evolution lab. I learned a lot about biology, about molecules. I did too. A lot of coding, bioinformatic work, and eventually ended up having my own lab, affectionately known as the Lab of Oz. And so what we really do in my lab, from sort of a broader perspective, is to understand the rules that govern microbial evolution. Again, with the goal that understanding the rules will help us understand how life originated on Earth. And so why microbial? Well, let's zoom in on that pale blue dot that is our planet. Although majority of life that we can see with our eyes is macroscopic, microbes actually rule the world. There's 100 million times as many bacteria in the oceans as there are stars in the known universe. And you just heard from Helen in a previous talk that, you know, even microbes within us constitute a high proportion of our body weight. And so it's not only dominated, the Earth is not only dominated by microbes now, it was dominated by microbes back and at the time of early Earth history. And so most likely when life originated, it was microbial. So that's why we're focusing on microbial evolution. But of course, you know, they're also very cool to think about. Now, going back to change via mutation that I introduced you earlier, this is not the only way microbes evolve. The microbes figured it out that this is not the most efficient way to gain some new function. Instead, what they do, they share the genes that are already known to do something. And that's called horizontal gene transfer. And to do that, they have all sorts of mechanisms. They can connect to each other using a variety of bridges that they create. And so through these channels, they literally can pass genetic information. So this is in a way similar to eukaryotic sex, but not mechanistically, just conceptually. But also, this gene exchange can occur via viruses. And so in the ocean, there are a lot of viral infections. You know, viruses are on our minds these days anyway, but think about this, that 20 to 40% of all bacteria in the ocean every day die from viral infections. And a lot of viruses known to carry genes that are not really viral genes. For example, just a couple of headline titles of the papers. For example, they can carry photosynthesis genes. This would be the genes that bacteria that can do photosynthesis have, that allow them to harvest the sunlight. But a lot of viruses carry those genes that they can prolong the life of the host, so it can photosynthesize. The same idea that phages or viruses, that's just bacterial virus lingo, bacteriophages, can spread antibiotic resistant genes because they carry them in their genomes and they infect different bacteria, including those in our gut. All right. So now I'm going to just focus on one project that is about a very strange bacterial virus. I'm very excited about this project. And let me introduce you what this strange virus is. We're going to think about a specific bacterium called Rhodobacter capsulatus. It's an aquatic bacterium, lives in lakes. Its relatives live in oceans. And so something funny happens when these bacteria are crowded together. About 0.1 or 3% of the cells in this crowded population start to undergo transformation. They turn on about 24 genes that previously were turned off. And when they turn on those genes, they start to produce what looks like viral particles. So this is an electron micrograph of what they produce. They're very small nanoscale particles. So it looks like viruses, virus-sized. And then unlike viruses usually package their own DNA so they can propagate and replicate in another bacterium. These guys don't. They carry a random piece of Rhodobacter capsulatus, the bacterial DNA. And then they kill the bacterium that produces them. And then these particles get released to the environment. And what they can do, they can deliver this DNA to other cells by infecting them, but there's no viral genome in there. So the question is, what is this? They were dubbed gene transfer agents because they're not really viruses, but it looks like they can transfer genes, so GTAs for short. And so in my lab, we're studying what are these GTAs, what are they for, and how did they evolve? And so what are they for? Well, they could be for a variety of functions. And so here This is just a cartoon illustration of hypotheses. For example, they can help other members of the population to provide genes that fix DNA damage that could be caused by UV light, or maybe, you know, they find the population of this bacteria find themselves in an environment where only some of them have some genes, others don't, so they can obtain new useful traits. By the way, this is a side project of my undergraduate student to make a children's book about this project, and we're in the process of trying to find a publisher. So I'm using some of her artwork here, Emma Esterman, who just graduated. So these are just hypotheses. They need to be tested, but these are possibilities. And so what we do know that these GTAs evolved from what was regular virus in the past some long time ago. So the question is, when did this happen and how did this happen? And so this is where we can use our archaeological techniques that I introduced to you earlier to try to figure this out. And so let me step back and talk a little bit about conceptually how we do that. And let's imagine you have an ancestral DNA sequence, and over time it starts to accumulate changes. So the changes here are shown in yellow, and it starts also to diversify. So you have now one variant that got these two changes. You get another variant that has these two changes. And now let's imagine that these are our contemporary bacteria, and this is their ancestor. Now, when we sample data now, this is a black box. We don't know what's happened, but we only have these two pieces of information, but we can model how these genetic changes came about. And there are a lot of models available, and I'm not going to go into details of them, but the idea is that using these models, we can reconstruct events that gave rise to these sequences, and then we can see what happens. All right, so again, this then would lead to this tree of past events connecting the contemporary genomes. That's just a picture that I showed you earlier. But now what you look here is the actual data. Ignore the labels, it's just different names of related bacteria. The Rhodobacter capsulatus is within this wedge here. And so what we've done, we looked at DNA sequences similar to those of Rhodobacter capsulatus and asked the question where the GTAs are. And we find that everything shown here in green, there's this big group of related organisms that actually have GTAs. And it looks like, just based on inference, if they all have it, the ancestor must have had it. So somewhere here, we think it originated somewhere here in the past or earlier maybe. And the other technique that, again, I cannot today go into details, but we can use fossils and other evidence to actually date when this happened. It's not always very accurate, but we can put some kind of a range, which is, you can see here, between 800 and 1800 million years ago. So pretty wide interval, but quite ancient if you just even look at 800 million years ago. So this GTA has been around for a while. And so this is not just some kind of a new defective virus. It's maintained in a pretty large group of organisms, so bacteria. And what we know from evolution of microbes is that if they were not using this, they would have gotten rid of it. It's expensive to maintain useless DNA in microbes. They try to get rid of things. They're very minimalistic. And so we think these GTAs are actually maintained in these genomes. And so the next thing that we've done, we actually looked at amino acid compositions of proteins that make up GTAs and compare them to viruses. And what we found that they're actually very different. So what I'm showing you here is just an example of one gene, doesn't matter what it is, but we just basically counted how many of the 20 amino acids that are known to be used by any organism on Earth are present in this viral protein versus GTA protein. And we just color code them here. So you can just by eyeballing see that the color bars for GTAs versus viruses are different. And I'm not going to show you here the data, but we actually demonstrated that this difference is statistically significant. So they're not just some kind of viruses that somehow just weird ones. So there is a difference, and so this difference is so pronounced that we can actually use a machine learning techniques to train our models, machine learning models, to distinguish GTAs from viruses just by looking at the amino acid composition. So we developed a program in the lab called GTA Hunter, and so basically this program now, if you give it a gene that maybe GTA, maybe viral, it will spit out the answer, what it thinks it is. And by applying this program to many bacterial genomes, we actually found a lot of sequences that potentially are GTA. So we think GTAs as a phenomenon is much more widespread than we think it was. All right. So now this change in amino acid composition is also not random. It turned out that this amino acids that GTAs use is skewed towards energetically cheap amino acids. And here again, I'm showing you a piece of the data for the same gene that I showed you before. The way we quantify what energetically cheaper means is how many carbons are in amino acids. So the more carbons there is, the more expensive it is to produce an amino acid. And if you have more amino acids with larger number of carbons in a protein, it's more expensive to make that protein. And so what you see here is how many carbons per amino acid side chain is present in GTA on average versus in viruses. And you can see here the viruses have many more than GTAs, and it's actually statistically significant, and we can show it that it's true for other genes as well. So this analysis then allows us to create a working hypothesis that GTAs are devices for survival under energy or nutrient limited conditions. Maybe this is when you want to have some new genes or have DNA repair happens. Now this hypothesis will need to be tested in the lab. In the actual experimental lab, we do computational predictions in my lab. So I'm trying to convince some collaborators to do some experiments. So so that is cool. We could take some computational analysis and make predictions that could be tested. Now there is another puzzle that is completely unresolved about this system, is how this system is maintained over time. Because if you remember what I told you earlier, the bacterium that actually produces GTAs get basically blown to release all those GTAs. So the question, if it gets killed, how do these genes actually survive in a population? What is a driving selective factor? This is a fascinating question to which I don't have an answer for right now, but we're working on modeling and doing some analysis for this. All right, so you know, this, a lot of people tell me this is very esoteric, and why would you study this? And is there anything about GTAs that could be useful for practical and societal application? And the answer is yes, there is. There is actually a company that is interested in looking into this system. It's far from being implemented, but we, in general, could use virus-like particle as a gene therapy, and it actually has been used as such. And so understanding in general how the genetic exchange occurs in something that looks like a virus, whether it's in bacteria or not, can advance our gene therapy efforts. But you can also think about treating of bacterial infections with engineered bacterial viruses. We right now running out of antibiotic treatments, and so maybe targeting and first of all designing bacterial viruses that carry basically something that can kill infectious bacteria, could be achieved via GTA-like particles. So just to summarize what I hope I conveyed in my talk. So I study computational biology. I like it because it, first of all, allows time travel and can also provide insights into biological puzzles that you can't necessarily get to by experiments, but I collaborate with a lot of experimental biologists to test our hypotheses. And in general, studying microbial evolution can shed light into rules of life, to more of a basic question about how life evolves. And then finally, more specifically to this project, acquisition and modification of already existing genes is an important evolutionary force. Microbes maintain many mechanisms for such gene exchange, and GTAs may be one of them. Now, in the final couple of minutes, I just wanted to say that a lot of the work that I was presented was not done just by me, but by members of my lab. That included postdocs, graduate students, and a lot of undergraduate students. And what you see here in parentheses is where those students are, if they don't have anything in parentheses. They're currently in my lab. Otherwise, you know, they work in different places. A lot of undergraduate students went on and into PhD programs. Specifically, Danielle and Taylor and Camille worked on that GTA Hunter program. They actually designed it from scratch. They over years we worked on this. Emma Esterman, she did not just book, but analyzed a lot of genes from GTAs. And currently, Jonathan and Jack continue to work on this GTA project. Now, the other thing that I want to mention, that in my lab, I have both computer science and biology majors working. Depending on the project, they either focus on the coding part or more biological interpretation part, but they get to interact with each other and you know, learn both disciplines. And here's just thank you for a lot of funding. So with that, I can entertain some questions if I figure out how to get out of this. I think I can stop sharing. And thank you for your attention. All right, I think if you put things in the chat, I can read them. I think we're okay on time, right? Sort of.
Yeah, we have about 5 more minutes. To anyone watching the live stream, I know there's a bit of a lag, but we're also moderating the live stream. So if you have any questions, you can pop it in the YouTube chat as well. Everyone's very non-inquisitive this morning.
I have one question actually. Since I'm a CS person, how can I, how can I get involved in like research like yours once I'm in college, right? What do you recommend for CS majors to be doing?
Yeah, that's a great question. Actually, you know, a lot of times I just get inquiries. So I think as Helen was saying in her previous talk, a lot of times it's just you need to be proactive, and you need to find a person whose research is of interest to you, and contact them. And usually I talk to a lot of students, and if I have openings, my lab is pretty small, but you know, students graduate, and so if I have openings, I usually chat with the students and see what kind of common interests we have, where do they want to get with this project, and we discuss possible projects. It's usually a kind of, it's not me telling you, okay, if you want to join my lab, you're going to do X. It's more like, hey, what are you interested in? What am I interested in? Let's find, you know, what can we work together on? So that's kind of an advice. Just find a professor who does something that is of interest to you and reach out. And you know, if one professor says no, you know, there are multiple reasons why that could be. It's not about you. It could be maybe, you know, they already have 5 other students. So don't be discouraged and keep asking several professors. All right, I think there are some questions. I'm going to start with the first one. When were GTAs first discovered? Oh, that's a great question. They were actually discovered back in the 70s, I think 1975. Nobody paid a lot of attention to them. They were kind of obscure for a while because people thought like, oh, this is weird. Okay. And I think a lot of, at that time, there was just not a lot of genomic data. Well, none of the genomic data was available at that time. And I think we are right now in biology in this unprecedented time when we can just look at the genomes and infer a lot of stuff that was not available. It was just harder to study. So right now GTAs are getting more traction because we can see them without actually seeing them in the cultures. Do you take a high school student? And the answer is yes, it depends. A lot of times, again, it's by contact. We do have programs here for high school students. So most of the time, if I will take a student, I will take it through the program just because this way it's the students will get additional experience, not just, you know, working in my lab, but also getting other professional development advice. Would you need any lab experience before joining a lab as a curious high schooler? Well, I usually, you know, I get a lot of inquiries, and of course, you know, I cannot take all of the students. I usually end up with taking 1 or 2 summer students, for example. If you know how to code, it's a plus, because a lot of time, everything pretty much what we do in the lab requires, like, for example, Python coding. And it's hard for a very short, say, summertime for you to start from scratch and just not knowing anything. So a lot of times I would definitely give a preference to a student who already knows how to code, then they can come and work on the project a little bit, you know, more meaningfully. Question from YouTube live stream. What are some COVID-proof research project ideas for high school students? Anything that doesn't need a lab or going to a public place? Well, so I haven't been, you know, right now I'm not at Dartmouth and I'm in my house in rural Vermont. All of the work that we've been doing, including what I presented today, have been done remotely. My graduate students and my undergraduate students are working remotely. We have computer servers to which we connect, so all the work is done on those servers. We communicate via Zoom. We haven't been to the lab. We have all our meetings, group meetings over Zoom. We work like that. We use Slack as well to communicate. So yes, a lot of computational work is actually done virtually, and my genomics class this fall will be done virtually as well. All right, looks like this is all for the questions. All right, well, thank you everyone for tuning in.
All right, thank you so much, Dr. Zhaxybayeva. I hope everyone really enjoyed her talk and learning about GTAs and you know, bioinformatics and computational biology.